Gary, the screen is still on the computer, but it's off on the board. Everybody, and welcome to our 10th annual Hampton-based Science Research Symposium. We would like to give a special welcome to Mr. Christopher Bouchard, our high school principal, Mr. Michael Morgan, our high school assistant principal, Mr. Michael Carlson, our assistant superintendent for curriculum, instruction, and assessment, Mr. Eric Ferraro, our executive director for educational services and school safety, our board of education members, and to all of our family, friends, mentors, and members that are in the audience this evening. Tonight, our research students will take you on a journey through their research on topics such as growth of Toxoplasma gondii, the effect of immunotherapy on cancer, and various materials that can be used to mitigate space radiation. We will begin with verbal presentations by each of our sophomore, junior, and senior science research students. To conclude the night, there will be poster presentations by all research students, freshman to senior, throughout the atrium to the auditorium and throughout the cafeteria. I would like to introduce Mr. Michael Carlson, our Assistant Superintendent for Curriculum, Instruction, and Assessment.
Good evening, and welcome to the Hampton-based High School's 10th Annual Research in Science Symposium. We're so glad to have all of you here as our guests tonight. As we begin, I want to take a moment on behalf of the Board of Education and the entire school district to say congratulations. We know that this journey and the work that has gone into tonight has been challenging, to say the least. But we also know, as John F. Kennedy has said, you didn't choose to do this because it was easy, but because it was hard. And tonight, we celebrate your hard work and the path that lies ahead due to your unending determination in finding the truth. The purpose of tonight's symposium is to meet the sophomores, juniors, and seniors who are deeply engaged in their scientific research, and to enjoy our presentations, which for some represent the culmination of 4 years of work developing and pursuing a scientific question. Tonight, you get to hear about the journey of science research, the methods that have led to discovery, and the way that each project will contribute to the scientific community that reaches far beyond the walls of our high school and of Hampton Bays. What we celebrate tonight is not just the culmination, but the process, the scientific method. One that started with an observation or a noticing, that led to a question or a wondering, and how powerful that initial thought must have been, and how equally difficult it must have also been to not jump to a conclusion without designing the method of experimentation, collecting empirical data, before finally deriving a conclusion. Science is a discipline, and scientists are disciplined, avoiding the temptation to fix or alter the truth, and speaking for nature by letting nature speak. Our program is unique, and as mentioned before, you'll see and hear how our students have already reached well beyond our classrooms to engage in this society of explorers through reviewing peer-reviewed articles, contributing to data collection, and some even having clinical experiences. Earning acceptance into this society of explorers is not something that's easily earned. It's reserved for those that are not only well-studied, but also for those who understand the research and the struggle that comes with it. It's done for the greater good, and the reward is the contribution of knowledge and its use by others. Every step along the way of scientific research is critically important and challenging. The Science Research Program is the most challenging course of study that our students in Hampton Bays can take. This work is done over 4 years in small groups, individually with Mrs. Lavazzoli, with a mentor, and with a tremendous amount of independent study. Under the very best conditions, this is rigorous and a challenging 4 years. Throw in sports, maybe a job, the college application process, and all the other things that a high schooler needs to navigate, and the road becomes that much more challenging. As you will see tonight, our young researchers are committed to the work and continue to rise to the occasion. I'd like to also acknowledge some special guests who have long been supporters of our science research program. Board of Education members Kevin Springer, Rich Joslin, Doc Capuano, Ann Culhane, and Liz Scully. New York State Assemblyman Fred Thiel, Suffolk County Legislator Bridget Fleming, Southampton Town Supervisor Jay Snyderman, and Town Board members Rick Martell, John Vovier, Tommy John Giovanni, and Cindy McNamara. Southampton Town Youth Services Director Lynn Nillis, and Assistant Director Tracy Colson. Once again, I'd like to acknowledge the mentors in the field and from around the nation that have willingly committed to our young student researchers. You will hear from 13 students tonight, presenting their work, each at a different point on the scientific research road, with all 23 students within our program giving poster presentations following this portion of the program. As scientists say, a good day of research leaves you with more questions than answers. So although tonight reflects the culmination of some students' work over 4 years, we know many will continue their work as they enter the next level of education. It's been a privilege to see how their work has developed throughout the years. Finally, I'd like to credit the university and the high school program at the University at Albany and its directors. We value this tremendous partnership with you and appreciate your commitment to us and your willingness to help us innovate. Beginning in the summer before their junior year, these students begin earning college credits through Albany, which amounts to 12 credits by the completion of their senior year. I want to offer a special thanks to Mrs. Mary Beth Lavazzoli for her coordination of this program and all the members of the science department from kindergarten through 12th grade. Thank you all for being with us tonight. Enjoy your time at Hampton Bays High School. And remember that you're always welcome here. And we hope among us that there are younger, budding scientists that leave here inspired to find the answers to their questions through this research program in the future. Our students are ready to introduce you to their research work. Ladies and gentlemen, enjoy hearing these young people as they present their work, which many will say is also their passion. I'd like to now introduce Dr. Angelica Dries, who I had her son when I was in Port Jefferson. From Brookhaven National Lab, who will speak about her work with the heavy ion polymer.
Hello, good evening, everybody. Oh, does it work? this one? It does. Thank you for having me. So this is a first for me to speak into a microphone. And if you've ever spoken at events or something, it is a 101. Keep eye contact. Because these fights, this is really hard. I have to say, so I will do my best and try. So tonight I'm going to tell you a little bit about where I work, which is Brookhaven National Laboratory. I even brought a little, pin, for you, and how we do some recycling, how we do use supercool magnets, and we do all this to study the Big Bang. So this is where I actually come from. My interest was in the universe originally, and I also wanted to make you a little bit proud of being a Long Islander. Long Island is, I don't know, has more to offer than just beaches. Even though they are, of course, nice and beautiful, and the infamous Hamptons, but it also has Brookhaven National Lab and some research instruments that are really cool. And we, we scientists, we come literally from all over the world. All right. We will figure this out as we go. So you see me here in the tunnel of my accelerator with colleagues. Of course, I'm not doing this alone. We are several hundred needed to operate what we are doing. So I'm personally from Germany. That's why I have this funny accent. I'm a physicist. I tried to figure out all your teachers here, but there are no real physics teachers. So that's really too bad. However, there are some of you that have a test tomorrow, so just that you know, there will be questions about, about what I'm presenting now, in the test tomorrow. So I worked at CERN, Geneva, Switzerland, before I came here 25 years ago, and I specialized in particle accelerators, and I'm going to tell you a little bit more about them. So, why particle accelerator? It sounds probably kind of random to you guys. I don't know. So I actually, when I was younger, and people wanted to become, riding a train or something like that. I wanted to be an astronaut, and go to the moon. And this all happened before you guys, but then there was this Challenger disaster, when it blew up and people actually died, and I was like, oh, no, I'm not going anymore. So I said, okay, so instead of going to the moon, I studied the origin of the universe from afar. So you see a picture here of Brookhaven National Lab, right here on Long Island. Again, so it's, we are quite an international group there from people, from Europe, from China, from Russia, from Japan, from America, the US as well. And just to give you an idea, this is my accelerator that you see up here. I'll tell you more about this one. There is another one down, you see that down here, which is called a light source. And my building, just to have a little bit of an idea, it's a pretty large area. It was a military camp, Camp Hampton. I don't know if you've ever heard about this. This was actually a military camp in World War II, and it maintained the charm of the military camp. So the buildings are not necessarily very nice, but practical. So let me talk about a little bit about recycling and the history that we have on the Brookhaven Lab. We had a program in the early 1980s where, which was called Isabel, and where we wanted to build a 3.8 kilometer long ring, that you see here. So this is actually, drawn on top of a photograph, and we started building the tunnel, which you can kind of see here. So this is during its construction, and it was actually canceled, in 1983 because what we planned there wouldn't quite work. And then it kind of sat there for a while, and then we decided, what else can we do? with that tunnel that already existed. So you can actually see here, this is that same tunnel from the inside. So we use that same ring and the buildings that we have here actually. You see these white squares up there, this is the buildings where we have detectors in there, and we use that to build RIC, which is what we are running now. To this day, we started it in 1999. So it's like 16 years, in between those. So it takes quite a while to design something, this size, and build it and put it together. So there are two of those rings in there, both 3.8 kilometers long. And we are going to use the same tunnel, you figured, and actually also the same magnets that we built here and that are working. And I'm going to use them for the Electron Ion Collider. That's what EIC stands for, which is again the same ring, but we will have 2 more accelerators in there. So right now we have 2, then we will have 4 of them, again in the same tunnel. So we're making, I don't know if you're taxpayers yet, but you will be soon. We are making the best use of the taxpayers' money here because we are all funded through the Department of Energy and try to, be very conscientious about what we have. So the name RIC, what does it stand for? R stands for relativistic, HI for heavy ion, and C for collider. In order for anything to collide, you need 2, right? Or else, that doesn't work. So accelerators often come only with 1, but it won't be a collider then. So we have 2 rings, those, we call them blue and yellow. Physicists are actually kind of funny, you see that, they're trying to make jokes, so we make the blue and yellow, and where they go into one, beam pipe, we call this green, because blue and yellow makes green, and things like that. Relativistic just means something very, very close to the speed of light. I don't know if you are familiar with Einstein, you can't quite reach the speed of light, but close enough. In our case, this is like 99.9995% of the speed of light, which means it goes 80,000 times, in 1 second around the 3.8 kilometer long range. So whatever you do, you're going to be too slow. So everything has to be computer assisted. So what particles do we collide? I don't know. So I spoke to chemistry teachers, so you must have had some idea about chemistry. You have heard the word atom. Somebody said atom splitter earlier. I forgot who that was, but I think your English teacher. We don't split atoms. Actually, what we use is only, parts of the nucleus of atoms, or the nuclei themselves. And, so mostly we use protons. Sometimes, together, with protons and neutrons forming a nucleus. And, inside actually a proton and a neutron, there are 3 other particles. Have you ever heard the word quark? Have you? So, actually, all my emojis didn't make it, so there was a funny face here, but anyway, so, the physicist, Murray Gell-Mann in 1963, named, all these quarks up, down, strange, charm, top and bottom, it's like, again, so it's the physicist's sense of humor. Anyway, so these particles or heavy ions, or nuclei of gold, for instance, or uranium, is what we have in those 2 rings, and we collide them. So, and what we need in order to get them on a circle, we need magnets. So, or else, once you are at the speed of light, you would go straight, right? You wouldn't go on a circle, So you have to tell them somehow, please turn here, and keep turning and turning so that, you keep going around and around. So we need magnets. So, within these big pipes that you saw, there are actually magnets on the inside, and we use very special supercooled magnets, we call them superconducting. The reason we use superconducting magnets is the following. while you actually have, normal wires, have a resistance. I don't know, have you ever changed a light bulb at home? I hope you have. I mean, if you touch them too soon after they burn out, we know that they are hot. So instead of just producing light, they also produce heat. They actually produce more heat than light. If something had no resistance, a wire had no resistance at all, you would use all of the electric current to produce what you want, light in this case, or a magnetic field in our case. So, instead of, you see here a light bulb, that produces light and heat. So the reason we did that is that we want very, very strong magnets because, again, those particles are heavy, they are fast, speed of light, they need to, turn on those circles, so we need them super, super strong. And in order to have this no resistance in the wire, we need to cool them to a very, very low temperature. So that temperature here, where the resistance goes completely away, is just a few degrees Celsius, which is like the same temperature that you have in outer space. And so we get that with liquid helium. So just to give you an idea how these superconducting magnets look like, they were proposed for all three of the projects, Isabel, Rick, and the EIC. Again, the temperature is about -450 Fahrenheit. Me being from Germany, I never really know that, so that's why I have the number written here. It's just, it's just a few Kelvin, that's the unit that we use. And liquid helium, so in order to do that, when we start a run, we are about to start our accelerator again for 3 more years, by the way, before it gets dismantled for the next project. We get trailers, big, truckloads full of liquid helium from the offsite. We fill it in, and then it takes days, for this to actually cool down. So it's about 300 degrees Celsius, or again, you do the math. In Fahrenheit, you will have that in your test tomorrow. In order for it to stay cold inside, so this is actually a picture from the magnet at the LHC at CERN in Switzerland, but the principle is the same. You have the beam pipes in the center, you have the magnets here, and surrounded, you have the liquid helium around it to keep it cool. And in order to keep everything cool inside and not outside, you need shielding and, this big vessel around it. So I prepared a little video for you so that you can have some idea what happens when those particles actually collide. The heavy nuclei was, the first idea that we had. Protons were something that we did a little bit later. We actually create something that is like a soup, the so-called quark-gluon plasma. And that quark-gluon plasma was last seen, in the universe right after the Big Bang. When I say right after, I mean like less than 1 second after. So this is something that hasn't been around for 15 billion years. And I think this is pretty cool that we can actually now create this and study this, which was the whole purpose of it. This now starts. So you see the particles here going from the so-called injectors into another injector, getting faster and faster as we go, and then actually collide. And you see the nice quarks that I mentioned, kept together by the gluons in order for both of them to make the quark-gluon plasma. They go through each other. And we actually thought that it would behave like a gas, so the quark-gluon plasma, that's why it actually has the name plasma. And it actually turns out that it's more like a liquid, and it behaves like a liquid. So that's why you see those two comparisons here, how it would look like if it was gas-like. And there should be another one, that is actually behaving like a liquid. And that was something that was actually a surprise, you know, and that's what science is, you know. When somebody said, I mean, like, Mike Carlson said, you shouldn't have foregone conclusions or expectations. You have to be open to how things can be different than what you think it should be. And to give you a little bit of an idea, you know, how we actually look at those particles that we then created in these collisions, you know, I brought you a picture of Captain Picard, just to give you a scale, you know, how large these, you know, devices, you know, you can call it a camera if you like, but this is, you know, like a 2-story house, you know, the size of one of those. This, of course, is just a computer, you know, digital picture of what they look at when something happened here at the center, a collision. You know, there are thousands, you know, up to 10,000 new particles are created out of the 2 colliding nuclei, and they leave all these traces in the detector, and that's a computer graphic that they look at as we go. But this is the actual detector, it's a photo of it, you know, so that takes, you know, those 10,000 particles, you know, several, you know, thousands times in 1 second, and then we run 24/7. Just to give you an idea, you know, when we run around the clock, you know, 24 hours per day, 7 days a week. There's no breaks, you know, we keep going. And so by now we have collected billions, you know, so I don't know if you wonder about this, but I would, you know, and why would you do this billions and millions of times? But there are some processes that we want to look at that are extremely rare, and you have to actually create, you know, many, you know, millions or billions in order to find them, you know, in your data set. So, you know, to wrap this up, I don't want to stress your attention span too much. So this is how we recycle tunnels and use supercooled magnets, you know, in several generations by now to study the quark-gluon plasma, which is my way of looking back at the Big Bang and the beginning of the universe. And that's really what my interest was and what I wanted to do, and that's my way of contributing to this. Thank you. Have questions, please?
Our first presentation of the night will be from junior Emily Barnes, whose research is on whale acoustics.
Hello, and thank you for being here tonight. My name is Emily Barnes, and my research is on whale acoustics. In the past years, my research focus has mainly been on passive acoustic monitoring, or PAM, and this is different types of PAM. I'll show you the picture. Some common types are buoys and tagging on whales and hydrophones coming from boats, which is all types of PAM monitoring. Recently, though, I've been studying more compact whale songs and whale sounds because I have started working on a project with my mentor where I analyze compact whale songs. Oh yeah, these are the two most common types of data, like the 2 most common types of data collectors that we get our data from for my project recently. We get them from buoys and gliders, and they both have hydrophones in them in order to be able to get the data from the whales, that when they sing and they make sounds. And these sounds can be a number of things, including grunting and humpback whale songs, but they're also used for a variety of purposes, and we aren't entirely sure what the purpose is always. If they can be used for mating and just communication in general, but we aren't entirely certain of the purpose of these songs in this current time. My mentor is Julian Wilder. She is really great, and she has helped me so much with furthering my research. She meets with me every 2 weeks, and then we go over any questions I have about bibliographic research, and she helps me with my presentations. And also, she's helped me see if I have any questions about my live data analyzing. She's affiliated with NOAA, and she is a cetacean acoustician, where she studies mainly whale songs, and she analyzes them from the spectrograms that we have in order to possibly gauge what type of whale they are and how far away they are. Recently, I've been performing a lot of bibliographic research on humpback whale songs in order to gain a better understanding for my project and how to analyze my data. The picture is a bottom mounting buoy, which is a type of buoy that we are also getting our data from for the live data stuff. And the whale songs are picked up by the PAN monitoring systems, which are in the buoy, and it also goes through satellite, which it bounces off, and then it comes into an archive in NOAA or in other places, like the Woods Hole Oceanographic Institution. Recently, two of the articles I have read are Where, When, and Why Do Western North Atlantic Humpback Whales Begin to Sing by Kowalski et al. And this really helped me understand a good example of different types of analysis, and it helped me understand some things about how they analyze different whale songs and why they sing, why they could possibly sing. I also read recently Acoustic Properties of Humpback Whale Songs by O et al., which was about, it helped me understand some properties and how they can be translated in the spectrograms, which I will show you later, about, so it can help me with my research. In the picture, it shows the buoy that we usually get data from, and the glider too, and a humpback whale.
Got it.
Recently, I'm working better to understand the sounds of humpback whales and to identify them from humpback whales from other types of whales, and also if they're a theme or not. And a theme is basically a repeated snippet of song that is over and over again. In this picture, it also shows another example of buoys and gliders, for your understanding. Currently, I am analyzing data from the humpback song themes, and the humpback song themes are repeated snippets up in the image, which shows time on the x-axis and frequency on the y-axis. It shows about a minute of elapsed time, and it shows that that humpback whale songs are very low frequency, and in the image, the types of notes and the frequency they are at are all shown. And this is an example of theming from my project, which shows a repeated pattern over and over in multiple songs, and it can be seen here. And this was from Martha's Vineyard, Massachusetts, the Massachusetts buoy that I've been mainly focusing my research on recently. In the future, I plan to continue with my bibliographic research and to continue to analyze humpback whale songs with my mentor. And my future and current research question is that which themes are present in each season of the Martha's Vineyard buoy deployment. And my 2 seasons are December 2021 through June 2022, and December 2022 through January 2023. This is when the deployment ended, and I haven't gotten any other data to switch it with, but I might do another buoy from Atlantic City too. And then we're going to see which themes are present in both, and if they're the same, if they're different, and if so, how different. Thank you.
The next presenter is Senior Caitlin Pinocchio. She's presenting her final research project on the removal of dehydrogenase enzymes in Tokoplasma, Golgi Grove, and Gene Expression.
Okay, can everybody hear me clearly?
Hi, my name is Caitlin Pinocchia. Thank you all so much for coming out here tonight. I hope you're looking forward to all of the future presentations. Tonight I'm presenting on my topic of Toxoplasma gondii, which is a human parasite. And my project is basically identifying genes within specific metabolic pathways that code for growth. So basically looking at genes that are important for growth of the parasite. These are two images of the parasite, and they're just fluorescent images. This is my mentor, Dr. Ira Blader. He's affiliated with the University of Buffalo, and he's a professor from the Department of Microbiology and Immunology. He has been a crucial key part to my research, and he's met with me so many times in the last couple of weeks. And I believe he's on virtually tonight, so I just wanted to give him a special thank you and shout out. He also directs research on Toxoplasma, as well as acts as a mentor to other students just like me, just on the undergraduate or graduate level. He facilitates and runs something called the Blader Lab, and the primary research question of the Blader Lab is what causes Toxoplasma growth, and that goes along with my project where I'm just trying to identify genes that are important for Toxoplasma growth. I chose this image because one of the factors that the lab looks at is neuroimmunology, and that looks at the way that the immune response of the brain reacts in response to a Toxoplasma infection. And I thought that was pretty interesting. Another thing that they do that I also am interested in is drug target discovery research. And they do this in ways, this is trying to figure out ways to kill the parasite to eliminate it from reproducing further. That brings me to what the parasite is. Well, it's considered a eukaryotic protozoan parasite, and it can be ingested through undercooked meat or cat excrement. I'll get into that later in the presentation. So it is a eukaryotic cell, and which means that it has a nucleus, which is similar to our DNA. So the cells function similarly to the cells in our bodies. And it is a protozoan parasite. So protozoan means that it's a singular celled organism. So the entire organism itself is just one cell, opposed to ourselves as an organism, we have billions and trillions of cells. It has something called an obligate intracellular lifestyle, which I'll get into further in the presentation, but that basically means that the parasite is not able to grow outside of a cell. And it can be estimated that 30 to 50% of the global population is infected with Toxoplasma, and up to 11% of the United States population is infected with Toxoplasma. And this is important because once you are infected with Toxoplasma and are exposed, it stays in your body for the rest of your life. So once you're exposed and you are infected, the Toxoplasma and the parasite stays in your body and suffer from the symptoms forever, unfortunately. That brings me to how Toxoplasma works. So basically you can ingest it through undercooked meat or cat excrement. So if you have a pet cat and you're cleaning out its cat litter and then you don't wash your hands and then you eat, you can ingest a cyst, which you can see it's pointing to the arrow. And if you digest the cyst, it can move to your brain and become, start off as a dormant cyst and then become reactivated and causing more severe symptoms as time goes on. This is what the Toxoplasma organism looks like when on the inside, and it actually has a lot of similarities to our cells. Like I said, it has a nucleus, so it codes DNA similarly to our organisms, or similar to our cells. But I find it interesting because it has two functions that our cells do not have. One of the functions being that it has the ability to enter a host cell, which is something that our cells can't do because our cells cannot enter other cells. And the other thing that I think is the most interesting part of this entire parasite is that it has the ability to manipulate a host cell's behavior. So once it's in the cell, it has the ability to manipulate the host cell's behavior, and it does things like stealing nutrients from the host, as well as other things. That brings me to how toxoplasma interacts with its environment. Toxoplasma lives within a parasitophorous vacuole, so that's where it lives within the cell. And you can see in the image, this is the nucleus, and then there's about 8 parasites actually living within this cell. And like I said, it's an obligate intracellular lifestyle, so it can only grow within the cell. And it grows by scavenging something called carbon sources from the host. So all of us have carbon sources within us, and that's what helps give us energy and helps us survive. So when it steals those nutrients, it's able to grow from us. So there's 2 different types of toxoplasma. The first type is tachyzoids, and this is less severe, and except they grow very fast and they cause disease. But the more severe version is called bradyzoids, and this is the version that exists within tissue cysts. They grow very slowly, and they're highly infectious, and this is where most of the problems lie. Toxoplasmosis is the main condition of toxoplasma infection. The main symptom of this being encephalitis, which is inflammation in the brain, and symptoms of encephalitis can vary from mild headaches to very severe things such as seizures and permanent brain damage. This image is actually taken by the Blader lab, I believe, and you can see that there's about 4 cysts or lesions within this patient's brain suffering from a toxoplasma infection. This is my review of literature. These articles have all been given to me by my mentor, and these have basically all helped me organize my project, understand my database, and figure out what I'm doing with this whole project. That brings me to my hypothesis and research question, and my hypothesis is that there are genes present within the central carbon metabolism that are important for toxoplasma growth, and I know that's a mouthful. The central carbon metabolism can be linked to almost all cellular metabolic processes of life. There's a total of 3 pathways within this metabolism, them being glycolysis, the TCA cycle, as well as oxidative phosphorylation. But in this project, I focused only on glycolysis and the TCA cycle. The image on the right is kind of just showing what a metabolism might look like within a human body. That brings me to my methodology. So my first step was to list out each step of the pathway in a pathway map. And then I broke down, I used a database to break down what genes encode for which enzymes. And then I asked a couple the first step of the glycolysis pathway at the top, it's a sugar glucose being broken down by hexokinase and being converted into glucose 6-phosphate. Hexokinase is where the gene lies, and this is the protein that's being coded. I selected a tab called transcriptomics, and this brought me to the graph on the right showing the differences in expression levels. And you can see that the expression is higher in tachyzoids and lower in bradyzoids. And like I said, bradyzoids are where the problem lies and where the tissue cysts are. So you can, since the expression is lower in bradyzoids, then it's somewhat unimportant for toxoplasma growth. That brings me to the second question that I asked, which was, what determines if a gene is important for growth or not? So then I selected a different button, this time called phenotype, and it took me to a graph that looked like this. You can see that the blue dot is in the middle, and if the blue dot is in the middle, then that means it's not important for growth, and I'll get into why. So if the blue dot is above the black line, star next to it shows that it's important for growth or not. You can see that there's about an even and you can see it actually looks a little bit different than glycolysis. Glycolysis had a variety of different colors. This one is mostly actually only expressed abundantly in tachyzoids and not really as much in bradyzoids or tissue cysts, but there are still plenty of genes found within the T. In the carbon source, the gene lies, then you can attack how the parasite is growing. That brings me to my future studies. I actually plan on continuing my research in the field of immunology next year at either the University of Albany and majoring in biology, or the SUNY Plattsburgh and majoring in biomedical sciences. That pretty much concludes my presentation. Thank you guys all so much for coming out here tonight. You guys are what makes it all possible. I wanted to give another special thanks to Ms. Lamazzoli. This program has really been taken so far, thanks to her. And yeah, have a good night.
Our next presenter is sophomore Isabel Hospital, presenting on the habitats of hypophobias.
My name is Isabel Hospitali, and I'm presenting habitats. Many researchers are trying to come up with ways to help preserve the piping plover habitats. Multiple articles I have looked into this year brought up the Endangered Species Act. This act provides a framework to conserve and protect endangered and threatened species and their habitats both domestically and abroad. They have also brought up creating barrier islands along Fire Island beaches to help preserve the areas where sea level rising destroys dunes and the plovers nesting. Ending the This year, I've spoken to two potential mentors, Dr. Christy Wills and Dr. Chelsea Weisman. Both Dr. Christy Wills and Chelsea Weisman are associated with the Virginia Polytechnic Institute State University. I've had the opportunity of meeting with both of them, and they've agreed to help me with my further bibliographic research. We had our first mentor meeting on March 23rd, where I informed them of the Hampton Bays Science Research Program, and they were able to help me and give me articles based off of what I told them. I have looked into some of the articles and had another meeting with them on April 19th, where I gave them a short summary of what I got out of the articles and asked some questions that I had regarding the plovers. The article I'm choosing to focus on tonight is Growth of Two Atlantic Coast Piping Plover Populations by my mentor, Chelsea Weisman. The Atlantic Coast Piping Plover, which was listed as endangered over 30 years ago, has not yet met recovery goals. They use a logistic exposure model, which expresses the daily survival rate as a linear combination of the covariates to estimate plover nesting daily survival rate. In the article, they monitored 196 nests throughout the Fire Island beaches. The estimated growth for Fire Island was greater 4 out of the 5 years, meaning that the plover population within the years will grow. As you can see in the graph, there was a huge spike in 2012 after the Hurricane Sandy. For the future, I plan to look into how vegetation on beaches affects the piping plovers and their habitats. I also would like to set up more meetings with Chelsea and Christy and figure out what they have been doing with their research and in their lab. Yeah.
Our fourth speaker tonight is Junior Dylan Foley, who is researching the ecology of horseshoe crabs.
Hello, my name is Dylan Foley, and this year my science symposium presentation is going to be on the ecology of horseshoe crabs, or Limitless polyphemus. Throughout this year, my research focus has been on the conservation of horseshoe crab habitats through beach nourishment programs and how environmental issues impact horseshoe crabs. This presentation is going to explain horseshoe crab behaviors, environmental stressors such as environmental stressors can exist in tidal height zones. Tidal height zones seem to have an effect on developing horseshoe crabs. The shoreline is divided into 3 zones, high, middle, and low, as shown in the image above. And the beach is divided into 3 spawning sites in the study by Christina Vasquez. Horseshoe crabs nest in the upper high tidal zones as is more optimal for development, while lower tidal zones may lead to delay in development because of low temperatures and hypoxic sediment, low oxygen, and may contain hydrogen sulfide. Luckily, horseshoe crabs have the ability to detect hydrogen sulfide and hypoxic levels. It was found in Christina Vasquez's study that temperature can accelerate or decelerate development, but risk from high temperature is higher, and the study also found trilobite were more tolerant to hypoxia. Another piece of research I've been conducting is on beach nourishment programs. The article I wrote by Nancy Jackson brings up the topic of coexistence as their title. And usually avoid places with high energy and low salinity. Many horseshoe crab habitats exist with the human settlement that ultimately habitat. The scale above shows the processes and responses that impact the suitability of habitats for horseshoe crabs, spawning over a spatial and temporal scale. These processes can impact habitats by changing shape, location, and sentiment. Management must Last year, I also had the opportunity to count, measure, and tag horseshoe crabs with the New York Horseshoe Crab Monitoring Program in 2022. I had the opportunity to talk with one of the site coordinators and other participants about the exciting topic of horseshoe crabs. My plan is to participate in the New York Horseshoe Crab Monitoring Program again for 2023, and to learn more about how to conduct my own research for my senior research project by first starting with testable questions. I also plan to participate in Project Vesta over the summer. As seen in the image above from last year's Project Vesta, Project Vesta was a beach nourishment project that dredged North Sea Harbor to replenish a local beach in North Sea. The natural mineral algae was added on top of the nourished beach as a natural way to sequester carbon dioxide and offset ocean acidification by increasing alkalinity. I would be involved in helping to determine if there was adverse effects to trace elements or byproducts of chemical weathering by algae on horseshoe crab eggs and other intertidal species. My focus is also to keep up with more bibliographic research. Thank you, and I hope you enjoyed the presentation.
Our next presentation of the evening will be given by Junior Emma Halsey, who will be discussing her research on sea turtle cold stunning.
Hello everyone, my name is Emma Halsey and I will be presenting my research topic on marine cold stunning on specific sea turtle species. The reason behind my choice in this area of research is my interest in marine ecosystems and finding ways to stop the negative impacts that have commonly occurred in these environments. The main theme of my research is very important to me because this is a huge issue that has contributed to the increasing mortality rates in marine habitats. Strandings cause sea turtle species to become lethargic and vulnerable to this marine problem such as cold stunning. Recent cases relating to hypothermia in sea turtles have commonly come up in the late fall and early winter months. Researchers are mainly focusing on chem-shielded species that climate change has an influence on increasing cases relating to sea surface temperatures as they have shown to continuously increase in waters. Some recent areas of study consist, again, of the effects rising water temperatures have on marine environments, and how specific habitats can result in affected tolerance levels for sea turtles. For example, this is a graph shown from one of my peer-reviewed articles I've read that shows the chem-shielded cold sun count versus the minimum of daily mean sea surface temperature from late October to early November. As you can see, both axes are expanding, which shows this is a negative data collection. Similarly, this is another graph from the same study that shows the chemistry cold stone count based on the predicted future minimum of daily mean sea surface temperature. This was also collected in late October through early November. This is in a study that predicts in the future the idea as sea surface temperature increases, cold stunning count will as well. This past July, I was lucky enough to establish a mentorship with Dr. Lucas Griffin, who is affiliated with the University of Massachusetts Amherst. Dr. Griffin specializes in environmental biology. My mentor provides me with documentaries and some of his own research, such as one of his many peer-reviewed articles titled Warming Seas Increase Cold Stunning. Marine Rescue Center that's affiliated with the Long Island Aquarium in Riverhead. I've taken animals and allow rescue centers to rehabilitate them, hopefully back to their natural state. About once a week, mostly in the winter season, I would go down to Ponquag Beach and patrol the area to see if there are any affected sea turtles on the beach. As of now, I have not personally found a stranded sea turtle, but I have heard of many recent cases of ones found at the rescue center. Again, within the cold stunning topic, there are many researchers and scientists questioning the connecting factors as to why this problem has been occurring so often. Through my research and peer-reviewed article readings, I have learned that these hypothermic-like behaviors could be caused by a lot of significant broad factors, including, again, as I said, warming sea surface temperatures, climate change, and just not having enough time to migrate out of waters. Water temperatures are dropping way too In the future, I want to further my bibliographic research through peer-reviewed articles that will extend my knowledge on the research topic of cold stunning. I am most interested in finding further articles that will help me gain a better understanding of the factors that influence migration patterns and efforts that will help decrease all these occurrences. Being able to extend my knowledge on climate change-induced factors such as wind, temperature, and salinity will also help me gain a little bit more background information about the causes of this problem. My future focus, again, also consists of being able to find some hands-on experience in a laboratory. Having this experience will allow my research to come from a more personal standpoint and a unique perspective, which I think is really important. As of right now, me and my mentor are in the process of developing a potential research question that I'm excited to assess in the future in my senior year. This concludes my presentation. Thank you for listening.
Our next presenter is sophomore Christopher Olden, who will be sharing his research on whale.
Hello, my name is Christopher Roldan, and this is my presentation for my research topic of science research. I was very interested in researching this area and topic because I like whales. And I'm also fond of the idea of discovering something new about a species in order to save it from being extinct. I've been focusing on many different areas of research, but recent research has been focusing my recent potential mentor that I've taken an interest into. He contributed to multiple publications that I found interesting, including a study on to what extent to which whales can remember certain locations and disturbances. One of the articles that Goldboken had contributed to was this article. This article was Oceanic Giants Dance to Atmospheric Rhythms, Ephemeral Wind-Driven Resource Tracking by Blue Whales, which was published on August 14th of last year and remains one of Goldboken's most recent works. Their initial hypothesis before doing this article was that whales sense disturbances in ocean currents in order to detect prey. The purpose of this article was to gain a better understanding of the way whales detect and find their prey, as many studies overlook this aspect of blue whales. They conducted this study through the dependency of acoustic sensing, and they only targeted blue whales for this study. They looked at blue whales and nothing else. These were the directions and magnitude of the acoustic sensing that they used to perform the experiment. You can see by diagram number C at the bottom left that this experiment was performed off the coast of Año Nuevo, and diagrams A and B display the acoustic sensing they use and the type of frequencies that worked best for them. They gained a lot of good results from the study, as they discovered that blue whales do indeed sense disturbances in the circulation of ocean currents to detect their prey, which was their initial hypothesis for the beginning, so they proved their hypothesis. Advance my research in the future, I would like to continue to build up my general understanding of the characteristics of whales and how we can save them using adaptive conservation methods. I would also like to get in touch with people enlightened on the subject, like Jeremy Goldboken, and I would like to hopefully conduct my own research in the future. Thank you for your time.
The seventh student presentation of the evening is Senior Sofia Solano Cruz, the salutatorian of the class of 2023. She will be presenting her senior research project on comparing the impact of cancer immunotherapy and distant metastasis melanoma on different age groups.
Good evening, everyone. My name is Sofia Solano, and this evening I will be talking to you about my research in cancer immunotherapy. So you might be wondering how exactly I got into this topic and into the science research program. Well, from a very young age, I've always had a very strong interest in science. I love to learn the why and the how behind everything I know to be true. And cancer in particular piqued my interest because Is one of those treatments that work throughout the body. It works in every cell in the body. And two of the medications that I have researched particularly over the past couple of years are nivolumab and ipilimumab. And they work similarly, but there are a few key differences between them. For example, nivolumab is a PD-L1 inhibitor, and it works on tumor cells. It inhibits the bonding between tumor cells and killer T cells, which are your body's immune cells that are responsible for killing any pathogens. These immune cells typically are in charge of killing things such as bacteria, viruses, fungi, anything that it identifies that can bring any sort of harm to the body. And tumor cells typically disguise themselves as healthy cells, so they typically go unnoticed by the immune system until unfortunately it's too late and nothing can really be done. So nivolumab binds to this protein and it inhibits the pathway that would make an immune cell ignore a healthy cell, and thus it allows the immune system to target this type of cell as something that needs to be eliminated and something that needs to be kicked out of the body and destroyed. And ipilimumab is a little similar to this. It works in a different pathway called the CTLA-4 pathway, and that's specifically on the T cells that are in charge of killing the bad cells or the bad pathogens in our body. And this pathway also inhibits an interaction which would make the immune cell stop killing something. So this interaction enables the immune system to continue to kill whatever cancer cell, tumor cell, it identifies as having this pathway that it needs to then destroy. And of course, none of this would be possible without my goal is essentially to compare the impact of cancer immunotherapy in distant metastasis melanoma patients less than 50 years old and 65 years or older. Goal, I've done a lot of independent research, bibliographic research more specifically. I have used a lot of what Dr. Snow has sent me, and I've also done a lot of my own research. And to back up my bibliographic research, I have also done a lot of data collection through the SEER database, and that is run by the National and everything I have discovered in my research and how I've accomplished my research goal. So here we can see a little bit of the data that I was discussing previously. This is the survival data for patients of distant metastasis melanoma. And we can see here the blue line represents the patients that are less than 50 years old, and the blue dots do as well. We can see the red line represents the patients that are older than 65. And we can see that both groups have an upward tendency. Both have increasing survival rates over the past 20 years or so, which is wonderful to see because, of course, we want patients to be surviving and have an increased quality of life. We want to see that upward tick, and we want to see that our current treatments are working, and that's what we are seeing. We see that about the year 2010, that's when the survival data really shows a good uptick in survival for distant metastasis melanoma patients. And that can partially be attributed to immunotherapy because that's when it began to be more mainstream. It began to be not just an experimental treatment, but something that was actually used more widely to treat patients. So we can see a big uptick in survival for both groups. But it's not until we look at the mortality data that we really see that older patients were more greatly benefited from immunotherapy. Oh, sorry. So we see here that it's like cutting it out a bit, but we see that the older patient group had a dip in mortality around the year 2015, which again can be partially attributed to these innovative methods of treating cancer through immunotherapy. And we see that the younger group has a relatively stable mortality rate. It's very near zero. However, a big limitation in my study is that the mortality rate, we can see here on the y-axis, only goes up to about 15%. Now you may be saying, wow, that's really low for mortality data for cancer. You would think it would be much higher, especially for distant cancer. Well, unfortunately, I wasn't able to find data specific to distant metastasis melanoma. The data you're seeing here for mortality is for melanoma in general. So you may be familiar with someone who has skin cancer. It's typically detected in the earlier stages, whether it's a mole or some other malformation on the skin. It's typically easy to detect and easy to remove through surgery because those are the beginning stages of cancer, stages 1 and 2. In those stages, it's more easily treatable and thus the mortality rate is much, much lower. And for distant metastasis melanoma, the mortality rate is significantly higher because of the fact that it is something that is spread throughout the body, as I mentioned previously. So we see here that the data isn't accurate. However, despite its inaccuracy, we do see that downwards trend in melanoma mortality. So we can see that despite the limitation of my data, this data still demonstrates that immunotherapy has had a positive response in patient outcomes and efficacy, safety, all of the above. So to discuss a little bit about my data, we can see both graphs demonstrated increased survival, which is wonderful. We love seeing that. We love seeing that quality of life is increasing, and we love seeing that people are dying less of a disease that was once almost considered a death sentence. That despite this, there is still a dip in the mortality, which indicates that treatment is effective even if the data is skewed to a lower mortality than what we were expecting. To sum up my presentation, immunotherapy has been a huge breakthrough in science, and we can see that in that despite its young age, it has been a promising cure to so many people that are suffering from distant metastasis melanoma and so many other cancers that have metastasized throughout their body. Thankfully, immunotherapy has less adverse side effects than certain medications such as chemotherapy or radiation, so we can definitely see that positive outlook, that positive aspect in cancer treatment, which brings us all a lot of hope. And it's especially gratifying to see that older patients are more positively affected than younger patients, because older patients, as I discussed previously, are more prone to becoming seriously ill or dying. So it's definitely a very hopeful outlook to see that the most vulnerable population is the one that's being most benefited. And we can see that survival has increased in both groups. So we're seeing that everyone is tending to live longer and have a better quality of life post-diagnosis than they did say 10, 15, 20 years ago. That concludes my presentation, but before I walk off the stage, I would like to send a huge thank you to Dr. Forsberg, who was my science research teacher for 3 years, and Ms. Lavazzoli, who took over the program this year. Both of you have been immensely helpful in my research, and I'm so grateful to have had you in this program. You guys have really made this program something that has greatly impacted my academic career in the most positive way possible. Thank you.
Our next presentation will be given by junior Emily Robinson, who will be discussing socioeconomic disparities and incidence of dementia.
Hello, my name is Emily Robinson, and tonight I'll be talking to you about the impact of socioeconomic disparities on the incidence of dementia. As displayed in the image, is the cortical thickness of 3 different groups, these being Alzheimer's disease, traumatic brain injury, and healthy aging. As you can see in healthy aging, there is, as you can see in healthy aging, those with healthy brains have the thickest cortex, whereas patients with Alzheimer's disease exhibit pronounced cortical thinning. Accelerated cortical thinning is illustrative of a higher risk of neurodegeneration and Alzheimer's disease-like trajectories. What my current research has been focusing on is how dementia incidence is influenced by both level of cognitive function and rate of cognitive decline later in life. One factor that is associated with this incidence of dementia and a decreased level of cognitive decline is low socioeconomic status, which can be proven by emerging data that indicates that both early life and late life socioeconomic status predict late life level of cognitive function. There is also evidence that Black people have a greater burden of lower socioeconomic status measures, such as a lower level of education, literacy, financial adequacy, and family income. This, as seen by the graph, results in higher prevalence of cognitive impairment among Black people. My mentor is Dr. Indira Turney, who is a postdoctoral neuroscience at Columbia University Medical Center. She is a part of the TAB Institute for Research on Alzheimer's Disease and the Aging Brain. This model shows the relationship between different measures of socioeconomic status, cognitive outcomes, and confounders. It shows how childhood socioeconomic status can impact early adult socioeconomic status, impacting late life socioeconomic status, which then can affect cognitive outcomes. There's also relationships where each stage of the life course impacts cognitive outcomes independently. For example, childhood socioeconomic status can impact cognitive outcomes independent of early adult and late life socioeconomic status. The C's are the confounding variables that impact each stage of the life course. C1 are the pre-childhood confounders, C2 are the post-childhood confounders, and C3 are the post-adulthood confounders. This figure shows that individuals with a stable low socioeconomic status at all time points had the quickest rate of decline, while at each time point, individuals with stable high socioeconomic status had the slowest rate of cognitive decline. This suggests that education is the most important socioeconomic contributor to the level of memory function, while while the late life income is the most important socioeconomic contributor to rate of cognitive decline. This means that while estimates indicate that high school education has the biggest impact on cognitive reserve, late life income appears more important for the rate of cognitive decline. This Cox proportional hazard model shows that Black participants have a greater hazard ratio for dementia than white participants. As stated before, Black people have a greater burden of low socioeconomic status. Another image showing differences in brain aging. This image examines racial and ethnic differences in cortical thickness and white matter hyperintensity volumes in midlife and late life. As you can see, Black-white disparities were larger than Latinx disparities for cortical thickness and white matter hyperintensity volume, suggesting that Latinx and white adults' brain aging was more pronounced in later life than in midlife, whereas Black adults showed accelerated brain aging starting as early as midlife rather than late life. In the future, I hope to look more into how to deconstruct the social determinants that drive disparities in brain health, as there's currently a lack of research in this area. Because education was the main contributor to level of cognitive function, interventions earlier in life may be necessary in order to increase cognitive reserve. On the other hand, because late life income predicts level of the rate of cognitive decline, interventions later in life may be necessary in order to slow the rate of cognitive decline. This suggests that both early and late life interventions may be necessary in order to delay dementia onset. In addition to this, there's also a need for interventions that will reduce and eliminate racial inequalities in cognitive and brain aging. This can possibly be done by improving the overall health of disadvantaged groups more rapidly than the rest of the population in order to eliminate inequalities. Taking all of these gaps of knowledge on current research into account, I was able to come up with my own research question, which is, what is the role of life course SES, childhood versus adulthood, in observed disparities in brain aging within different race and ethnicity groups? I hope that soon I'll be able to start finding answers to this research question while being able to collect data that I can start analyzing. That concludes my presentation. Thank you.
The next speaker of the night is Junior Manal Naeem, who will be presenting on various cardiovascular magnetic resonance imaging techniques.
Good evening, my name is Manal Naeem, and today I'll be discussing cardiovascular magnetic resonance imaging, or CMR imaging. CMR is a non-invasive imaging method that allows you to look at specific areas of the heart as well as analyze the function of the heart. This means that no instruments are placed into the heart. This is different from other methods such as CT scans or X-rays because it doesn't provide any harmful ionizing radiation. This imaging method allows identification of certain diseases, such as the one being shown here, which is hypertrophic cardiomyopathies, which will be explained later on. Throughout the last few years, my research has focused on non-invasive cardiac imaging and cardiomyopathies, which is a disease of the heart muscle or the myocardium. Before I get further into my presentation, I'd like to break down the heart into 5 different parts. This includes the left and right ventricles, which is the bottom chamber of the heart, and the left and right atrias, which is the top chamber of the heart, together producing 4 chambers of the heart. There's also the coronary arteries that supply blood to the heart, the electrical system which allows the heart to pump, and finally the myocardium, which consists of a few different layers. The image here shows a patient with hypertrophic cardiomyopathy, and the white arrow is pointing to the thickened muscle. My mentor is Dr. Warren J. Manning, who is affiliated with Beth Israel Deaconess and Harvard Medical School. I established a mentorship with him in May of last year, and specific things. But if there's a lower spatial resolution, which is a higher surface area, it allows you to look at larger areas, which isn't as helpful. The most commonly used imaging techniques in CMR are Cine imaging, T1 and T2 mapping, and T2 star, and LGE imaging. Cine imaging looks at the motion and the function of the heart. The video here is of a patient with a normal heart. Here you can see the heart pumping and blood going in and out of the heart. Cine imaging repeatedly takes images of the heart, and here you can also see the myocardium and the left and right ventricles. T1 and T2 mapping allow tissue characterization, and T2 star shows iron deposition. LGE imaging is used to look at areas of fibrosis, which is scar tissue. There are a few different types of LGE, which includes late gadolinium enhancement and early gadolinium enhancement. This is when gadolinium contrast agents are inserted into the heart, and images are taken either 5 to 10 minutes after the gadolinium disperses, which is early gadolinium enhancement, or EGE. Or the images are taken 10 to 20 minutes after the gadolinium disperses, which is late gadolinium enhancement. The image here is of a patient with a normal heart again, and it's an LGE image taken. The wider areas are the areas of LGE, but if these were more prominent, then there would be more LGE. But since this is a normal heart, the areas of LGE aren't as prominent. A few other techniques include myocardial tagging, which shows the heart muscle moving. This is different than other imaging techniques because it's one of the only methods that allows you to look at only the heart muscle moving. The graph shows.
I would like to continue to research the role of fingolimod in the treatment of acute ischemic stroke and the potential benefits of using this over an established treatment for stroke. I would also like to examine the bradycardia side effects in order to research if there's any way to lessen this or if targeting different receptors might be more beneficial. I would also like to examine sex differences in response to treatment, as there are differences observed in stroke, and this suggests that different treatments may be needed. As seen in the image, often both males and females face moderate injury, with females often being slightly more significant and sometimes falling under severe injury. I've begun to research the synthesis of myriosin from Isaria sinclairi,
When immunosuppressant activity has been discovered within other plants and fungi, and I'm interested in how this activity is discovered within a plant and why that plant might hold it. That concludes my presentation. Thank you for listening.
Our final presentation of the night will be given by Senior Elijah Amos. He will be presenting his final research project on what materials can be used to mitigate space radiation.
Hello there. So nice to see so many beautiful, smiling faces in the crowd tonight. This really makes me so happy to see so many folks willing and ready to still learn. So tonight I'm going to give you all a presentation on space radiation, and specifically how to mitigate space radiation in outer space. And before I start, I'll just say that there are two different types of space radiation. Those being solid particle events and galactic cosmic rays, which I'll now refer to as SPEs and GCRs. What materials lead to the least amount of radiation exposure? And so Dr. Bonadori and I tested 4 different materials: water, aluminum, polyethylene, and polybutylene terephthalate, which is quite a mouthful, some refer to as PBT for the remainder of this presentation. And basically, we decided to test out polybutylene terephthalate because of its high boron content. Boron is known for having very positive effects when it comes to absorbing radiation and mitigating such harsh doses. And we basically compiled all our data into the effective dose equivalent. And basically, the effective dose equivalent is measured in sieverts. And what sieverts are, they are a measure of radiation exposure. And so we wanted to lower the value because NASA has imposed a career limit of 800 millisieverts to 1,200 millisieverts, while it is known that ionizing cancer, or cancer caused by ionizing radiation, can start as low as doses at 100 millisieverts. So you want the dose to be as low as possible. So for GCR maximum, we have water coming at 222.5 millisieverts, aluminum at 438.4 millisieverts, polyethylene at 210.6 millisieverts, and PBT to be. But this is actually fully expected due to aluminum being more of an antiquated technology. Aluminum was used during the space race during the 1960s to help protect our astronauts whilst they were going to the moon. But in the ensuing decades, we have tested out and discovered more composite materials that are much more effective at mitigating this radiation, such as polyethylene, which had the lowest dose here out of all the GCR max data. That is actually because polyethylene order more temporary events while GCRs are, like I said, omnipresent. And so for water, we have 11.34 millisieverts, aluminum at 7.20. Miracle composite material that would propel us into the next age of space radiation mitigation. It wasn't so, but that's how science is. Test out hypothesis, and eventually you'll find what's most effective. And aluminum was expected ineffective except in cases of SVE. That was the only exception. And water being effective along polyethylene. And I like to mention water because I've actually been skipping over quite some bit. Water is great when it comes to being a molecule. It's known for its properties of cleansing, such as toxins and radiation. But in regards to space travel, it is highly inefficient due to its high cost effectiveness and its weight too. So we can't really look at water seriously, but it's nice to know that it has this general effectiveness. And for future plans, I plan on finishing a research paper based on my studies that will be out by hopefully the end of next month or early into June. And then upon graduation,
the stage for a brief question and answer session. If you have a question for any of these researchers, please raise your hand and a microphone is going to be brought to you.
Awesome job, by the way. It's not my field, except for the very last one, radiation. Hello. Of course I was glad to see that. And some of the studies subject was so wild, I didn't really understand it all. So, it's like really, really awesome job. I'm glad to see this group of kids here. So my question that I had was, there was a presentation, actually I have three. Can I ask three questions?
By all means, feel free. Absolutely.
One, was about horseshoe crabs. I didn't quite understand what is the importance. I mean, why would we want to actually even look at them? And what's the context?
So horseshoe crabs are extremely important in the biomedical industry, fishing industry, and are used for piping plovers in the when they're migratory birds and they feed on horseshoe crab eggs. And so basically they're useful as predators, prey, just I guess like overall fishing industry, biomedical industry, their blue blood, and how it combats diseases. Okay.
So are they endangered?
They it's sort of there's very little information out there on the population because it's very difficult to record the exact number of horseshoe crabs in the world. So that's
what trying to find out if they are endangered.
Yeah.
Oh, okay.
That's why they have counting and horseshoe crab recording, spawning and stuff.
All right, thank you. So my next question was about sea turtles. Oh, you are next week. I'll hand you that. So the sea turtles, the cold stunning, and then, one of the reasons you gave was global warming. This sounds counterintuitive to me. It warms, temperature goes up, but cold stunning goes.
Yeah, so I thought the same thing when I first researched it. So basically, water temperatures so sea surface temperature is heightening. And basically, when we move into the fall and winter months, the temperatures drop so quickly that it just causes a hypothermic shock. So they do warm, but then cause like a counteractive temperature change.
Okay, thank you. And then I promise it's my last question. So there was about cancer survivors, cancer rates, and figure. Oh no, I'm just going down the line here. So there were two graphs. One was the survival rate. Went up in the group of below 50s and over 65. I'm not quite there yet. Thank God. And that had a slope, right? Which you fitted. I remember that. And then there was the mortality rate, which was flat. Isn't that a contradiction? How can the mortality rate be flat when the survival rate goes up? So it should go down? No? Anyway.
Yeah, that's a great question. And if you see the mortality rate, unfortunately, that was one of the limitations of my data. I wasn't able to find specifically the mortality rate for distant metastasis melanoma, which is like the stage 3 and stage 4 melanoma that are typically known to be more deadly, whereas the survival rates were specific to the distant metastasis melanoma. So that's why we can see that.
Different data samples.
A little bit, yes. Yeah. But within that larger data sample of the general melanoma, there is that population of the distant metastasis melanoma. It's just muffled by the other melanoma patients within that data sample that aren't exactly the same as the survival data. Thank you.
Thank you. I mean, awesome job. Really, so I'm thrilled.
Other questions?
So this is the question for the person who did on dementia. So in the very beginning, you mentioned how like normal aging caused it was normal, but then if you had dementia or a brain injury, it was just your brain was messed up. How exactly was it deformed? What happened to it?
So basically, in normal aging, there's a thicker cortex, whereas in traumatic brain injury or Alzheimer's disease, there's cortical thinning, which this causes accelerated brain aging. So that's why there's like a difference in the brain for Alzheimer's disease and normal brain aging.
Hi, my question's for the same person. I actually really loved your topic. Since it's such a systematic level on how socioeconomic issues impact, minority groups, I was wondering, like, your idea of how long a true impact will be made, as it's such a big, broad, issue, and you express how there's very little research going into it. I wonder, from here until 10, 20 years, do you think could have a substantial impact?
So I think that the impacts affect not only 20 years, but for the rest of your life, because socioeconomic factors starting as early as childhood can impact brain aging later in life when you're in your 60s and 70s. So I think that it practically impacts your whole life, and you need to these socioeconomic factors impact it throughout early childhood, adulthood, and late life. So yeah.
My question is for Cassidy, and it's what else might hurt the BBB other than a stroke?
So different lifestyles might affect the BBB. That could include smoking, drinking, stress can have an effect on it. Different diseases could also affect the BBB. I thought it was interesting, Lyme's disease might have an effect on it. Epilepsy, MS, which is multiple sclerosis.
My question is for Maya. You mentioned studying RAS in humans, but are there any animal models that also demonstrate the effect of music on any sort of motor skill?
I do know of one study where it was RAS was performed on mice, but other than that, I actually haven't heard a lot about animal studies. So if I find more information on that in the future, I can give that to you.
Hi, this is for the same person. Kind of a follow-up on that one, because I was wondering if you'd heard anything on the relationship between music and plant growth, because I've heard some interesting things.
I actually have not been looking into that, but if I do look into it in the future, I will get back to you.
Thank you very much, ladies and gentlemen. You guys can feel free to go and head to your posters. I'd like to thank everybody very much for listening to our verbal presentations and for participating in our Q&A session. The next portion of our evening is going to be the poster presentations given by all of our research students from freshman to senior year. Please peruse the posters and have some dessert. There's dessert in the cafeteria, some cookies and cake, coffee, all that stuff. Thank you very much.